Hello, in this video we're going to introduce white blood cells. So these are the fighting cells in the body, right? And they are the soldiers, right? So their genesis, we start off in the bone marrow, and we see that, as usual, the pluripotent hematopoietic stem cell, via activation it will divide. And our white blood cells, they come from precursors of the myeloid progenitor and the lymphoid progenitor. The lymphoid progenitor producing lymphocytes, which are these, and the myeloid progenitor producing granulocytes or polymorphonucleocytes and monocytes, right? Now, the function of leukocytes or white blood cells is to fight infection. And unlike red blood cells, they lack hemoglobin, though they do not have the oxygen carrying capacity. They have a nucleus. They fight infections and destroy cancer cells. They are the cleaners of the system. Right? They clean off these germs, pollutants, and so on. Right? They do this via mechanisms such as phagocytosis or direct cell killing and the production of antibodies, right? Antibodies, they will act as markers to help the body identify that there lies a virus, there lies a carcinogen, right? Now we classify them into three groups. I talked about the groups, and see that the granulocytes, they mature in the bone marrow and they constitute the highest percentage, that is 60 to 70%. The monocytes, they mature in the spleen, liver, and lymph nodes, also called the reticuloendothelial tissues. The monocytes, they mature in the lymph tissues such as spleen, lymph nodes, and thymus. They constitute 20 to 30%. Now this is the average approximates, and we see that the average total white blood cell count should be within this range, and the average is 9,000. And the highest of the white blood cells, we have neutrophils, and the lowest being basophils, right? In the granulocytes, we have neutrophils, eosinophils, and basophils, right? And the neutrophils, they measure 12 to 15 centimeter micrometers, and they are stained by neutral dyes. They have a multi-lobed nuclei consisting of 3 to 5 lobes, as we saw or here, right? And we see that their activation, we'll talk more about the activation of leukocytes, is shown on this diagram. They are first attached to these adhesive proteins on the endothelium of the blood vessel. They are found circulating in the blood, and they have their lifespan. And when they are circulating in the blood, if there is let's say an emergency which calls them, right, they are trapped, or they are adhered to the endothelium, then they are forced to be activated by chemokines, and they undergo a conformational change, and this conformational change activates them, and they move and are transmigrated. And we see that they are transmigrated via two pathways. There's the paracellular and the transcellular. Paracellular, they go between cells. Transcellular, they go through cells, right? And we see that these neutrophils, they phagocyze microbes and unleash their toxic granule contents, which digest foreign materials. They are killers, these neutrophils. They destroy, they ingest. They are just amazing, right? And some of the granules, they contain their primary and secondary. The primary one, which I noise of interest is defensins, and myeloperoxidases, and the secondary ones are lactoferrins and collagenases, right? Then we have eosinophils, which are bilobed, and their granules stained red to pink, right? And they play a major role in the defense against parasites, bacteria, and viral infections, and they are the ones which bring about the allergic response. So this allergic response is brought about when they release toxic substances which are sometimes innocuous or non-harmful to parasites but may harm the host, right? They release histamine, they release so and so, right? And we see that these substances, they bring about harm to the host but not to the parasites, right? We see that when they are released, one can increase in temperature and so on, right? And basophils, also bilobed, stained with basic dyes, and their granules stained purple blue, right? And they contain heparin and glycosaminoglycans, and their main function is immunomodulation. They regulate via releasing chemokines, cytokines, and they are activated by immunoglobulin E, right? So when they are activated, they release histamine and serotonin and so on, and in patients who have asthma, this histamine and serotonin, mostly serotonin, right? They induce vasoconstriction, which will lead to breathing difficulties and vasodilation, which increases vascular permeability, leading to flashes, right? So whenever you see someone who is red like this, these vascular flashes, it shows that basophils are active, right? Now monocytes, these are shapeshifters, right? And they are found circulating in blood, and when they are trapped in tissues such as the spleen, the brain, they become specialized. In the brain, there are microglia. In the spleen, they are just macrophages, right? So monocytes are precursors, right, to the mononuclear phagocytic system, which includes cells such as osteoclast, macrophages, and microglia, right? These cells, right? So these monocytes have abundant cytoplasm, and their granules give them a bluish gray color on the microscope. They carry out phagocytosis. We shall talk about phagocytosis and define it, and destroy microbes. They are also involved in immunomodulation, right? Now, chemotaxis, this is just the way in which we direct our immune cells, right? Via the release of chemoattractants such as tumor necrosis, factor alpha, interleukin 1, interleukin 2, and bacterial products, right? And we see that this chemotaxis, right, the attraction levels, they are different, and we see the concentration is highest at the source and less at the periphery here, right? It has a concentration gradient. So as the let's say the neutrophils are attracted, as they get closer, they get more and more attracted, right? And they get attracted also when there is opsonization, which is just tagging by these antibodies, right? and complement cells from the complement system. We shall talk about complement cells, complement system, inflammation in the upcoming videos, right? So this phagocytosis, by definition, is just cellular ingestion of offending agents or foreign agents which are not recognized by the body. They do not contain the mark of being off the body, right? And you see that the most of the immune cells which are of phagocytosis are neutrophils and macrophages, right? And macrophages, they are formed from monocytes. So what happens, we take our microbe via antibodies to help these neutrophils and macrophages identify and recognize that this thing is foreign. Then we phagocytize it, that is, we engouge it. Then we engouge it into a phagosome. Then from phagosome we infuse our digestive enzymes, so in the lysosome packet here. Then we now have degenerative products. We have neutralized the microbe, right? And we see that phagocytosis is increased if the surface of the particle, that is this microbe, is rough and it lacks protective protein and it is bound to antibodies, right? That's opsonization. Take note, that's opsonization. When you have a bound antigen to antibodies, or a marked antigen or microbe, it's opsonization. Now the activation, as we saw on the diagram of neutrophils, it's as follows. We adhere to the endothelium due to this gradient of chemoattractants, as we said in chemotaxis. We undergo a conformational change, we transmigrate to the attraction site, and we do our job, right? Now some of the conditions or abnormalities: we have leukopenia, which is abnormally low number of white blood cells. You see here the white blood cells are normal. In leukopenia there will be few, and these white blood cells can be neutrophils, lymphocytes, monocytes, eosinophils, and basophils, right? And people who have leukopenia may increase their risks of infection since they do not have the normal count of white blood cells, and cancer development, right? Because, as I said, some of the white blood cells, such as basophils, they act in immunomodulation, right? And what can be the causes of leukopenia? We have cancer treatment, that is chemotherapy. Chemotherapy drugs, some of them are immunosuppressants. Some of them, they reduce the activity of the bone marrow, which is the site of the production of white blood cells, right? Then we have lymphoma, which is cancer of white blood cells called lymphocytes. Take note, it's lymphocytes. We have specialized, it's cancer of the lymphocytes. And the causes, it can be immunodeficiencies, it can be autoimmune diseases, right? It can be also infections, where it will just hijack the system, and there will now be uncontrolled division of lymphocytes, right? Now lastly, we have this table here. So when we have neutrophils, eosinophils, which are low, it's called the penia, right? And if they are high, it's called the philia. And the penia, it can be in neutrophils, it can be due to splenomegaly, that is an enlarged spleen. So if there's an enlarged spleen, you see that the destruction of these neutrophils will be high. Since neutrophils have a lifespan, they have to be destroyed. Then the philia part, if we have metabolic disorders, right? it will lead to their high production, since they will be attracted to those abnormalities, right? Then eosinophils, it can be penia, they can be low due to bacterial infection, or they can be high due to pulmonary disease or any form of infection, right? Then monocytes, they can be low, that is penia, due to ingestion of corticosteroids, or they can be high, monocytosis, right? Due to myeloproliferative diseases, right? Such as those who have polycythemia vera, right? Then lymphocytes can be low due to HIV and AIDS. It can be high, that is lymphocytosis, due to viral infections, right? So that's all on the introduction. It was just an overview. In the upcoming videos, we're going to have extensive explanation of inflammation, HIV, AIDS, the complement systems, right? So more coming. Make sure to subscribe, like, and share if this video helped you. Thank you for watching till the end.